Welcome to today's video where we dive deep into the fascinating world of genetics, specifically focusing on Mendelian randomization and causation. This intriguing concept, Mendelian randomization, is a method used in the field of genetics that leverages the power of genetics to determine causal relationships between various factors. The name comes from the father of modern genetics, Gregor Mendel, who conducted pioneering research into the inheritance patterns of certain traits. Today, we use Mendelian randomization to help us understand how different genetic variants might influence health outcomes. It's like using nature's own randomized control trial to investigate cause and effect. This method has become an important tool in understanding the complex interplay between genetics and our environment. It's a fascinating area of study that has wide-ranging implications in areas like disease research, drug development, and public health. Now that we have a basic understanding of Mendelian randomization, let's delve into its core assumptions. The first assumption in Mendelian randomization is that genetic variants are distributed randomly at conception. Now let's delve into this. Picture this. You're at a carnival, standing in front of a game of chance. You have no control over the outcome. It's all up to luck. This is a fitting metaphor for how genetic variants are distributed at conception. Essentially, it's a genetic lottery. When a new life is conceived, it inherits a unique mix of genetic variants from its parents. This mix is determined purely by chance, and it's this randomness that makes Mendelian randomization possible. Why is this random distribution so critical, you ask? Well, this assumption ensures that the genetic variants we inherit are not influenced by factors like lifestyle, environment, or socioeconomic status. These factors, which can affect our health outcomes, are unrelated to the genetic hand we're dealt at conception. It's like the carnival game we mentioned earlier. The result isn't affected by who you are or where you come from, it's all about the luck of the draw. This randomness also means that genetic variants are equally distributed across different groups of people. Whether you're looking at a group of athletes or a group of couch potatoes, you'll find the same range of genetic variants in both. This distribution allows researchers to compare groups with different lifestyles, diets, or other factors, and to isolate the impact of these factors on health outcomes. But here's the real kicker. This assumption of random distribution allows us to use genetic variants as a sort of natural experiment. By comparing people with different genetic variants, we can explore how these differences might influence health outcomes. And because these genetic differences are random, any differences we see in health outcomes can be attributed to the genetic variants, and not to other confounding factors. So you see, the concept of random distribution of genetic variants is not just a fancy scientific term. It's a cornerstone of Mendelian randomization. It's the reason why this method can provide such powerful insights into the causes of health outcomes. Our genes are like a lottery at conception, and this random distribution forms the basis of Mendelian randomization. The second assumption is that the genetic variants are independent of confounding factors. Now, before we dive into why this independence is so vital, let's first unpack what we mean by confounding factors. Imagine you're baking a cake. There are numerous ingredients that go into it, and each one affects the final outcome. If you change the quantity of one ingredient, say sugar, it will affect the taste of the cake. But what if you also change the amount of flour at the same time? Then you wouldn't be able to tell if the change in taste was due to the sugar, the flour, or a combination of both. The flour in this case is a confounding factor. It confuses the relationship between the sugar and the taste of the cake. In the context of genetics, confounding factors are variables that influence both the genetic variant and the outcome of interest. They can muddle our understanding of the relationship between the two. For example, if we're investigating a genetic variant related to lung function, smoking could be a confounding factor. Smoking affects both lung function and potentially the activity of the genetic variant. So why is it important that genetic variants are independent of these confounding factors? Let's go back to our cake analogy. If we want to truly understand the effect of sugar on the taste, we need to hold the amount of flour constant. Similarly, in genetics, if we want to understand the impact of a genetic variant on an outcome, we need to ensure that there are no confounding factors muddying the waters. This is where the beauty of Mendelian randomization comes in. You see, the genetic variants we inherit from our parents are determined at conception, long before any confounding factors can exert their influence. This means that they are, in essence, naturally independent of these factors. And this, my friends, is the crux of our second assumption. So the genetic lottery not only happens randomly, but it's also independent of external factors. The third assumption is that genetic variants influence the outcome only through the exposure. This assumption forms a critical part of our Mendelian randomization journey, and it's all about the pathway that genetic variants take to influence our outcomes. Picture this. You're walking through a maze. There are several routes you could take to reach the end, but only one will get you there. This illustrates the idea behind this assumption. The genetic variants in this case are like you trying to navigate through the maze being our body. The end of the maze? That's the outcome we're talking about. The assumption proposes that these genetic variants can't take any shortcuts or detours to influence the outcome. They have to go through a specific path, referred to as the exposure. The exposure can be anything that the genetic variant influences, which then has a knock-on effect on the outcome. For instance, a genetic variant might affect how much of a certain protein our bodies produce, which in turn could influence our likelihood of developing a particular disease. For Mendelian randomization to work, it's crucial that these genetic variants stick to the path and don't go wandering off course. If they start influencing the outcome through other routes, it muddles the picture, making it harder for us to accurately determine causation. It's like, if you were allowed to take shortcuts in the maze, we wouldn't be able to accurately determine how long it takes you to navigate it. The same principle applies here. If genetic variants influence the outcome through other means, it could lead to biased results, and that's something we want to avoid in scientific studies. This assumption is really important because it helps to maintain the integrity of the randomization process. It ensures that we're looking at a clear, straightforward relationship between the genetic variant and the outcome through the exposure. So, as we delve deeper into Mendelian randomization, remember this. Our genetic variants are like diligent maze walkers, sticking to the path, influencing our outcomes only by way of exposure. In other words, our genetic variants can only affect our outcomes by influencing our exposures. Now that we know the assumptions of Mendelian randomization, how does it apply to real-world scenarios? Well, let's start by appreciating the fact that Mendelian randomization, or MR for short, is a powerful tool in genetic research. By using the principles of genetics and statistical inference, MSAR can help scientists make sense of the complex tapestry of genes and their effects on various traits and diseases. One of the most significant applications of MR is its ability to infer causality. Now, this may sound a bit heavy, but let's break it down. In traditional observational studies, it's often challenging to determine whether a particular exposure causes a specific outcome. This is because there can be many confounding factors that could muddle the relationship. But this is where MSAR steps in, wearing its superhero cape. It uses genetic variants, which are randomly assigned at conception and remain constant throughout life, to act as proxies for the exposure of interest. This random allocation of genetic variants is akin to a natural randomized control trial, helping to circumvent the problem of confounders. Let's take an example. Suppose we're interested in understanding whether increased body mass index, or BMI, causes heart disease. In an observational study, we might find a link between higher BMI and heart disease. But could there be other factors like diet or physical activity influencing both BMI and heart disease? Absolutely. Enter Mester. In this scenario, Mster would use genetic variants associated with BMI as an instrument. These variants are not related to diet or physical activity, only to BMI. Therefore, any association between these genetic variants and heart disease can be seen as evidence of a causal effect of BMI on heart disease. And it's not just about health. MSTR has been used in a wide range of fields. For example, in social sciences, MSAR has been applied to study the effect of education on socioeconomic outcomes. In economics, MSAR has been used to infer the causal effect of income on happiness. But what about the future? With the advent of large-scale genetic data, the potential for MR is skyrocketing. It can aid in drug development by identifying potential therapeutic targets. It can also help in predicting disease risk and informing public health policies. What's more, MSAR's ability to test multiple hypotheses simultaneously can lead to some exciting discoveries. For instance, it can examine the effect of smoking on multiple outcomes like lung cancer, heart disease, and longevity, all at the same time. Similarly, it can also look at how different exposures like diet, physical activity, and stress independently affect an outcome such as depression. But remember, while MSAR is a powerful tool, it's not a magic wand. It's crucial to interpret the results of MSTR studies with caution, considering its assumptions and limitations, which we'll explore in the next scene. In a nutshell, Mendelian randomization takes us on a fascinating journey. into the world of genetics and causality. It's like a detective, using clues from our genes to unravel the complex web of cause and effect. Through Mendelian randomization, we can get a glimpse into the complex world of genetics and causality. So as we delve deeper into the secrets of our genetic blueprint, who knows what mysteries we'll uncover next? Like any scientific theory, Mendelian randomization is not without its critics and limitations. This statement rings true for many scientific theories, and Mendelian randomization is no exception. As we delve into the limitations and criticisms of this intriguing concept, it's crucial to remember that no theory is flawless, and constructive critique often leads to its evolution. One of the key criticisms revolves around the stringent assumptions that underpin Mendelian randomization. The theory leans heavily on the idea that genetic variants are randomly allocated at conception and are independent of confounding factors. However, in reality, this may not always be the case. Genes do not exist in isolation, they interact with each other and with environmental factors in complex ways that can confound the results of a Mendelian randomization study. Another common criticism is that Mendelian randomization assumes that the genetic variants influence the outcome only through the exposure. This is known as the no horizontal pleiotropy assumption. Pleiotropy occurs when a single gene influences multiple phenotypic traits. When this happens, it can create a backdoor path that may bias the results. While statistical methods exist to detect and adjust for pleiotropy, it remains a challenge and a source of potential bias. A further limitation is the potential for population stratification. This occurs when there are systematic differences in allele frequencies between subpopulations within a population due to different ancestry. If not properly accounted for, population stratification can lead to spurious associations and false conclusions. Moreover, the interpretation of Mendelian randomization results can be tricky. It's tempting to view a significant result as proof of a causal effect, but it's important to remember that Mendelian randomization provides evidence of causality, not proof. It's just one piece of the puzzle in the quest to understand complex genetic relationships. Lastly, Mendelian randomization studies rely on large sample sizes to provide sufficient power to detect an effect. This can be a limitation, as it may not always be feasible to gather such large datasets. Plus, the results are population-level estimates and may not apply to individuals, limiting their clinical applicability. Despite these limitations and criticisms, it's important to reflect on the value that Mendelian randomization brings to the field of genetic epidemiology. It provides a way to investigate potential causal relationships in observational data, offering insights that might otherwise remain hidden. Moreover, it's an evolving field, and new methods are continually being developed to address its limitations. So, while Mendelian randomization may have its critics and limitations, it's crucial to remember that it's a continually evolving field. As we refine our methods and deepen our understanding, we can better leverage this powerful tool to unravel the complex relationship between our genes and our health. While not perfect, Mendelian randomization is a powerful tool in understanding the intricate relationship between our genes and our health. We've covered a lot of ground in this video, from understanding Mendelian randomization to its real-world applications and criticisms. We've learned how genetic variants are randomly distributed at conception, how they remain independent, and how they influence outcomes only through exposure. We've also delved into the practical uses of Mendelian randomization, from drug discovery to public health policy. However, we've also faced the limitations and criticisms head-on, acknowledging that Mendelian randomization is not a one-size-fits-all solution, but a powerful tool in the geneticist's toolkit. The journey into the world of genetics is filled with complexities and nuances, much like the genome itself. But don't let that deter you. Thank you for joining us on this genetic journey. Don't forget to subscribe, share, and comment below. You can also find the transcript of this video in the description. Stay curious, and keep exploring the world of genetics.